Abhinandan. I welcome cordially and heartily welcome the guest dignitaries on the dais as well as the people who are present at this occasion on this auditorium, in this auditorium as well as those who are connected with us. From the premises of Birsa Agricultural University, I welcome all the participants who are participating in this national seminar on crop breeding for vital adaptation. Once again, most welcome to the dignitaries on the dais, the Honorable Vice Chancellor, Birsa Agricultural University, Dr. O.N. Singh sir, as well as the other dignitaries on the dais. I welcome all of you today. As we all know, like always, we are here for the national seminar on crop breeding for wider adaptation. I request all the people in the auditorium to please keep the phone on the silence mode. आप सभी का तहे दिल से हार्दिक अभिनंदन। बिरसा कृषि विश्वविद्यालय के परिसर से मैं सभी पार्टिसिपेंट्स का जो आज ऑनलाइन हमारे साथ मौजूद हैं उन सभी का अभिनंदन करती हूँ। मंचासीन मंच पर हमारे साथ मौजूद माननीय कुलपति बिरसा कृषि विश्वविद्यालय डॉक्टर ओ.एन. सिंह साहब, साथ ही साथ अन्य अतिथिगण जो मंचासीन हमारे साथ मौजूद हैं आज के इस नेशनल सेमिनार में। As we all know, the dignitaries are present here, and as far as Corona and COVID-19 is concerned, we are attending this online national seminar. So I welcome all the dignitaries, all the participants who are connected with us today. As per our tradition and culture, we welcome the guests on this occasion, the dignitaries who are present on the dais. I would request the students to please come forward and present a flower to the dignitaries on the dais. For floral welcome, I would request the students to welcome the Honorable Vice Chancellor, Birsa Agricultural University, Dr. O.N. Singh sir, as well as the dignitaries on the dais, Dr. M.S. Yadav, Dean Agriculture, Dr. J.D. Hadar sir, Dr. D.P. Singh from DHU, Dr. D.N. Singh sir, he's also there, Dr. Thank you very much. This was the floral welcome to the dignitaries on the dais, but with that, I welcome all the dignitaries also who are not present with us today on this occasion. As per our tradition and culture, we always start the program after the lighting of lamp. दीप प्रज्वलन के साथ हर एक कार्यक्रम की हम शुरुआत करते हैं। माननीय अतिथियों से सादर निवेदन दीप प्रज्वलित कर आज के इस कार्यक्रम की शुरुआत करें। I request the dignitaries on the dais, Honorable Vice Chancellor sir is requested to lighten the lamp and inaugurate the national seminar. The dignitaries on the dais are also requested to please come forward for the lighting of lamp. दीप प्रज्वलन की परंपरा हमारी प्राचीन परंपरा रही है, अपनी संस्कृति का हिस्सा है। किसी भी नेक कार्य की शुरुआत दीप प्रज्वलन के साथ करते हैं, इस उम्मीद के साथ कि आने वाले वक्त में दीपक की प्रज्वलित लौ यह उजाला यह प्रकाश समाज को एक नई दिशा दे पाएगा, नई रोशनी दे पाएगा। जिस नेशनल सेमिनार में हम सब उपस्थित हैं, ज्ञान की बातें तमाम बातें यहाँ करेंगे। उम्मीद है इन बातों की रोशनी समाज के विभिन्न हिस्सों तक पहुंचेगी। इसी कामना के साथ, शुभकामना के साथ दीप प्रज्वलित कर आज के इस कार्यक्रम की विधिवत शुरुआत। मंचासीन हमारे अतिथियों के द्वारा, मंचासीन मंच पर मौजूद लोगों के द्वारा नेशनल सेमिनार में माननीय कुलपति बिरसा कृषि विश्वविद्यालय के द्वारा दीप प्रज्वलित कर आज के इस कार्यक्रम की शुरुआत की गई। बतौर मुख्य अतिथि माननीय कुलपति बिरसा कृषि विश्वविद्यालय डॉक्टर ओ.एन. सिंह सर, बहुत बहुत धन्यवाद आप सभी का आज के इस अवसर पर दीप प्रज्वलित कर आज के इस कार्यक्रम की हम सभी ने शुरुआत की है। नेशनल सेमिनार में मौजूद तमाम लोगों का तहे दिल से पुनः एक बार अभिनंदन। मंचासीन अतिथियों का दीप प्रज्वलन के लिए बहुत बहुत धन्यवाद। आज के इस अवसर पर हमारे साथ हमारे अतिथिगण मौजूद हैं। सभी लोग जो आज हमारे साथ यहाँ मौजूद हैं, हम तहे दिल से उनके आभारी हैं। अभिनंदन के शब्दों के साथ मंचासीन अपने अतिथियों से मैं अनुरोध करती हूँ हमारे बीच मौजूद डीन एग्रीकल्चर डॉक्टर M.S. यादव सर से कि वो आगे आए और यहाँ मौजूद तमाम लोगों का तथा तमाम अतिथियों का अभिनंदन करें। डॉक्टर M.S. यादव, डीन एग्रीकल्चर।
Good morning, Honorable Vice Chancellor, Birsa Agricultural University, Ranchi, Dr. O.N. Singh, Vice President of this Ranchi chapter, Dr. D.N. Singh, Secretary, Dr. J.D. Haider. I welcome you all in two-day online national seminar, that is webinar on crop breeding for wider adaptation. It is being organized by Indian Society of Genetics and Plant Breeding, Ranchi chapter, which covers the plant breeders' activity of the state of Jharkhand, Bihar, Uttar Pradesh, and West Bengal. Around 1,000 scientists are connected with it. I also welcome again all the scientists, Honorable Vice Chancellor Dr. O.N. Singh, and who is also a plant breeder and administrator at this time in Birsa Agricultural University. I welcome Dr. G.P. Singh, breeder and the President of ISGPD, New Delhi. I welcome Dr. Professor B.D. Singh Sahab, President of Ranchi chapter. I know him very well because of his name familiar to agricultural students, especially plant breeding students, for his book suited to PG and undergraduate students. I was really eager to welcome all the scientists related to GPB society. From actually last evening, I was very much eager to see them at Ranchi campus. Similarly, this Jharkhand is very beautiful, naturally created by the God, but something is between us and COVID. Otherwise, we have been here all together at this campus. I am thankful to Ranchi chapter of Indian Society of Genetics and Plant Breeding for giving me this opportunity to welcome you all. I thank Secretary Dr. J.D. Haider, Vice President of this chapter, Dr. D.N. Singh. We all agriculturists are fortunate to listen eminent plant breeders after this inaugural function. And the names are Dr. A. Singh, who is renowned molecular breeder, who is also Vice Chancellor of IARI, New Delhi. Dr. S.S. Bagga, who was also earlier, who has served us at this campus, and he will also give a talk. Dr. Kuldeep Singh was planned to give a talk, but Dr. S.R. Das, who is senior plant breeder, I think he is retired, will also give a talk for the benefit of all the breeders. Dr. N.P. Singh, Director IIPR, Kanpur, will also provide us a lecture on this topic. Thank you very much.
Thank you very much for your welcome address. The warm words of welcome has warmed our heart today, and now we are forwarding towards the national seminar on which we are present here today. As we all know that we all are working in our directions, specific directions, we are doing our work continuously, thanklessly, effortlessly, 365 days. But doing the work and compiling it and benefiting the other people by that is also one purpose for which we are doing our work. So this is a moment where I would request the person of this national seminar, the secretary who has organized the seminar, the person who is behind the seminar, to come forward and give us the secretary report. He is going to give the secretary report to the people about the work and the things going on continuously, not in the university but also in this subject. मैं रिक्वेस्ट करूंगी आज के इस अवसर पर हमारे बीच मौजूद जिन्होंने इस कार्यक्रम का आयोजन किया, निरंतर किया है, ISGPV के रांची चैप्टर में हमने कई सालों से इस कार्यक्रम को देखा है। ISPGR न्यू दिल्ली के सहयोग से हर वर्ष रांची में बिरसा कृषि विश्वविद्यालय में आयोजित होता है और निरंतर अपने कार्य के लिए जाना जाता है। इसकी बेहतरीन प्रगति में जिसका सबसे महत्वपूर्ण योगदान है, निरंतर इतने वर्षों तक जिन्होंने अपनी अहम भूमिका निभाई है, आज के इस अवसर पर वो अपने द्वारा किए गए कार्यों को उन कार्यों के माध्यम से हो रहे लोगों के लाभ के विषय में आज इस अवसर पर हम सभी को रूबरू कराएंगे। मैं मंच पर आमंत्रित करती हूँ सेक्रेटरी रिपोर्ट के लिए डॉक्टर JD हैरिस।
Thank you, Shashi. Respected Dr. Ranchi, Honorable Vice Chancellor and Chief Guest of today's function, Dr. B.D. Singh Sahab, who is presiding the session online from BHU, Dr. G.P. Singh Sahab, who is the President of Indian Society, who is also online, my colleague Dr. T.N. Singh, Vice President, Dr. Swahan Ram, Director Research, Dr. Revadu, DPG, Dr. Gupta, Dr. Pal, Dr. Shahi, Dr. K.K. Singh, Dr. Himanshu from Biotechnology, and all the members present here offline during this inauguration from the department. I welcome all the persons and I feel pleasure in giving some names who have been very cooperative to the society, not this year only, all the times when we ask them, approach them, they also helped us. I would like to name them. Dr. A.K. Singh, Director and Vice Chancellor, Indian Institute of Agricultural Research. Dr. S.S. Bangal, our colleague here in early days, National Professor, ICR, and presently he is in PAU Ludhiana. Dr. Kuldeep Singh, Director, NDPGR. Dr. N.K. Singh, from Biotechnology, Dr. N.P. Singh, Indian Institute of Pulsar Research, Director, Kanpur, Dr. S.R. Das, our senior and renowned breeder, Dr. Jambulkar from BAT, and other chairmen of the invited lecturers. I welcome all on behalf of the society. I feel pleasure in introducing first in few lines about the society. The Ranchi chapter, a satellite unit of most prestigious Indian Society of Genetics and Plant Breeding, New Delhi, since its inception on 28 August 2012, has been vigorously working to create the interest of students of all age groups towards fascinating science of genetics, plant breeding, and biotechnology through essay competitions, inviting the students from local schools, colleges in the Department of Plant Breeding and Genetics, Birsa Agricultural University, for their interaction on the subject and exposure to students and scientists of the university. At intervals, holding lecture seminars to provide platform for young scientists, PhD scholars to present their work before scientific gathering, interact with renowned scientists for knowledge sharing and collaboration in research. During the year 2019, the executive committee decided to involve and sensitize even younger group of school students through painting competitions. We were happy to see the creativity of this group, the students too. And in November last year, when we had to hold this seminar, we facilitated all the students of all age groups line with the objective of the parent society, the Indian Society of Genetics and Plant Breeding I am talking of, and Ranchi chapter, we are holding national seminars on topics that require timely attention to bring young as well as renowned scientists of the field at one platform, discuss the issues and come out with sound research strategies. In view of El Nino on Indian agriculture, the present national seminar on crop breeding for wider adaptation was originally scheduled on 9-10 November 2019, which for compelling reasons was deferred for March 22nd and 23rd, 2020. The stage was set for this schedule, but for COVID-19 pandemic, the organizers had to postpone it. During initial months of pandemic, everyone felt that normal livelihood activities will be difficult to come on track. Thank God, with the dedicated efforts of the government, the life by and large has come to near normalcy. The abnormal situation due to COVID-19 compelled to take advantage of available technologies. Using this web tech, we have virtually assembled to discuss the important issues related to crop breeding for wider adaptation tools. To achieve the objectives of the seminar, diverse themes have been structured so as to gather ideas of wider group and come to a meaningful and applicable strategies. And for it, we collaborated besides our parent society with Indian Society of Plant Genetic Resources New Delhi and NABARD had the support of all collaborators, Indian Society of Genetics and Plant Breeding New Delhi, Birsa Agricultural University Ranchi, Indian Society of Plant Genetic Resources New Delhi, and NABARD is acknowledged. I am sure we are going to be immensely benefited with the experience sharing of lead and invited speakers. The presentations of young, curious minds during technical sessions will also contribute for our future strategies. I wish all the best.
बहुत बहुत धन्यवाद। रांची चैप्टर के द्वारा आयोजित इस कार्यक्रम में तमाम बातें जो पूरे साल किए गए उन कार्यों का विवरण एक रूप में यहाँ पे दिया जाता है। संक्षिप्त रूप में उसे देना संभव नहीं है, फिर भी उसे संकलित करके आज एब्स्ट्रैक्ट के रूप में आपके समक्ष प्रस्तुत किया जाएगा, ताकि जो लोग यहाँ मौजूद नहीं हैं, जो इसमें शामिल नहीं हो पाए, वो सब भी उसे जरूर पढ़ पाए। उन सभी कार्यों को संकलित करके एब्स्ट्रैक्ट का रूप देकर आज यहाँ पर रिलीज करने का अनुरोध मैं आज यहाँ पर मौजूद हमारे माननीय कुलपति बिरसा कृषि विश्वविद्यालय डॉक्टर ON सिंह साहब से करूंगी। आज के इस अवसर पर जो रिलीज है वो है एक एब्स्ट्रैक्ट और साथ में एक पेंटिंग है। मैं माननीय कुलपति बिरसा कृषि विश्वविद्यालय डॉक्टर ON सिंह साहब से अनुरोध करती हूँ कि आज के इस अवसर पर इस एब्स्ट्रैक्ट को आज यहाँ रिलीज करें, जो नेशनल सेमिनार में प्रस्तुत किया जाएगा। I request the Honorable Vice Chancellor to release the abstract which has been compiled in this national seminar by the people who are doing their work continuously, effortlessly. They are working, and this has been compiled for the information to the people who are all around. So this is the release of the abstract. Along with this, we have some paintings which has to be released today. Now may I request the Honorable Vice Chancellor Dr. ON Singh sir to please release the painting at this occasion. In this national seminar, we all are here. This has been organized by the Ranchi chapter on crop breeding for wider adaptation. The scientists are effortlessly working all around the year in the university as well as in the whole country. And these are the releases which will reach to the people who are not present here or are not connected with us today. Thank you very much sir for the release. बहुत बहुत धन्यवाद माननीय कुलपति महोदय को आज के इस अवसर पर उन्होंने इसे रिलीज किया। जैसा कि आप सब जानते हैं कि यह ISGBB का रांची चैप्टर है और कंटिन्यूअसली इस माध्यम से यहाँ पे कार्य किया जा रहा है। संपूर्ण भारत के लोग इससे जुड़े हैं और साथ में आज के इस सेमिनार में सभी लोग मौजूद हैं। रांची चैप्टर अपने बेहतरीन प्रदर्शन कर करते हुए निरंतर अपने कार्य में लगा है। क्रॉप ब्रीडिंग फॉर वाइडर एडेप्टेशन के इस नेशनल सेमिनार में आज हमारे साथ मौजूद हैं वाइस प्रेसिडेंट इस रांची चैप्टर के डॉक्टर डी एन सिंह साहब। मैं उनसे अनुरोध करती हूँ उनके वक्तव्य के।
Honorable Vice Chancellor of this esteemed Agriculture University, that is Birsa Agricultural University, and also Chief Guest of 2 days National Seminar on Crop Breeding for Wider Adaptation, and Dr. M.S. Yadav, Dean Faculty of Agriculture, Secretary of the Ranchi Chapter Indian Society of Genetics and Plant Breeding, and Chairman Department of Genetics and Plant Breeding, Dr. J.D. Haider, my colleague Dr. Somadam, Chief Scientist Lindsay. I could see in the audience Dr. M.K. Gupta, Dean Faculty of Postgraduate Studies and also Director Resident Instruction, Director Research Dr. Vadhu, Dr. D.K. Sai, DSW, Dr. Paul, Associate Dean, College of Agriculture, Gadwa, and all the participating scientists. It's a really great honor and pleasure for me to be part of this national seminar on crop breeding for wider adaptation. And I want to, Mr. Chief Guest and Honorable Vice Chancellor, the kind of contribution which has been made by this Indian Society of Genetics and Plant Breeding. In 20th of century, if you recall back, this society, our National Society, Indian Society of Genetics and Plant Breeding, was established before independence, that is 1941, with the president, very first president, Dr. B.B. Pal, who was the first Director General of also Imperial Council of Agriculture Research before independence, and later on it became Indian Council of Agriculture Research. As the very first secretary, I think I'm very happy to name Sir T.S. Venkataraman. He was the first secretary, and Britishers had entitled him, given honor, that is Sir T.S. Venkataraman. So the first president, Dr. B.B. Pal, who happens to be the first Director General of Imperial Council of Agriculture Research and later on Indian Council of Agriculture Research, and then Sir T.S. Venkataraman, who was Director of Sugarcane Breeding Institute. These are the two people, rather many people who have contributed very significantly for strengthening the activities of Indian Society of Genetics and Plant Breeding, not at the national level but also global level, because this Indian Journal of Genetics and Plant Breeding, this is internationally rated, and nowadays NAS rating is more than 6. Sir, if you recall back the history and the kind of contribution which has been made by 20th century, I think two revolutions was the major breakthrough, that is first Green Revolution and second is information technology. And I think Green Revolution make this country from food deficit to food surplus, and information technology, if you recall back Dr. Sam Petroda, really he was the man, and late Prime Minister Rajiv Gandhi, they are the pillar for making this contribution to take place. And information technology that has made life very easy, and whole world has become a global village. And the kind of information which is flowing, especially in terms of science and technology, that has really reached to the very down, everybody who are the stakeholder are interested to know the knowledge and information. Now, coming to the kind of contribution which has been made by this geneticist and plant breeder, and also by the Indian Society of Genetics and Plant Breeding, as I said, that at the time of independence we were food deficit, and we were producing hardly 50 million tons of the total food grade, and we were importing especially rice and wheat from the other countries. And if you recall, the second Prime Minister, late Lal Bahadur Shastri, who has given a very popular slogan, that is Jai Jawan Jai Kisan, and that has happened in between 16 and 70, that is a Green Revolution. And this has happened that this year, 1920 and 2021, record production of rice, that is 117 million tons. Means at the place of 20 million tons of rice which are being produced at the time of independence, 6 times production and productivity has gone of rice, only because of integration of one dwarfing gene, that is DGO, which was discovered from the Taiwan. And the very first two variety of the rice, if you recall, the Sora Chaussat and Larma Rojo. I did not in case of wheat, but in case of rice, that is IR8 and Taichu native. These have contributed, and today we have more than 1,000 variety of rice for all possible ecology, and they have almost the blood of that actually given IRA. So that has contributed very significantly, and I said that rice production from 20 million tons has gone to 117 million tons. Similarly, with the wheat, neighboring 10, that has introduced and integrated in our land races and the cultivated variety. And the first two variety, Larmarojo and Sarvata Sonoro, they have introduced, and later on Kalyan Sona and Sonalika. And at the time of independence, only 9 million tons wheat were being produced. And last year, I think Dr. Surdas is sitting here, 107 million tons of the wheat record production. And 2018, Honorable Vice Chancellor sir, we had an annual group meeting of wheat and barley here under the leadership of Dr. Tilochan Mahapatra, the secretary there, and DG ICR. And he has given the target to the Indian scientists and also some cement scientists who are sitting, that is to produce 10 tons average productivity of the wheat. So I think there are some genotype, and this year, last year, we have harvested some of the genotype, have given 7 tons. I think the result which has been presented by the plant breeder here. So I think the kind of genotype which are really being bred by Indian Institute of Wheat and Barley, I think they are able to give, and also International Center of Maize and Wheat Improvement. They are developing a kind of genotype which are really giving 7 to 9 tons, 7 to 10 tons. So I think the wheat production, which was at the time of independence only 9 million tons, has gone 107 million tons. So this has become possible only because of the advancement of the science of genetics and plant breeding. And third revolution has taken place in maize. You see, Honorable Vice Chancellor sir, the hybridization of, in case of maize, and the production and productivity, and nowadays in US, where maize is considered as a crop, 100% that is cultivation of maize that is under single cross hybrid. And here in our country also, 90%, I think 9 to 10 million hectares, that is under the maize, and 30 million tons we are producing. So if you see, that is another remarkable contribution which has been made by the scientists of genetics and plant breeder. And the 7 million tons maize which were being produced at the time of independence, it has gone to 30 million tons only because of the developing hybrid variety in case of maize, especially the single cross hybrid. Now another fourth very important contribution, that is under the sugar cane. I was naming the name of T.S. Venkatraman. He was the director and breeder of Sugarcane Breeding Institute, Coimbatore. And earlier, whatever variety of sugarcane were being cultivated, that is Saccharum officinarum. But now, whatever variety that is being cultivated in sugarcane, that is not Saccharum officinarum, but it is a Saccharum complex. Means number of species they have introduced, and not only sugar content has increased, but total production and productivity. So these 4 major contributions which has been made by the scientists of genetics and plant breeding, that is DGO Uzain in case of rice, Norrington in wheat, and hybridization in maize, and then the sugar cane, that Sir T.S. Venkatraman, that these are the major achievements which has been taken place. Nowadays, the genetics, the topic is also crop breeding for wider adaptation. I think we are trying to develop with the help of the molecular plant breeder by marker assisted plant breeding and marker assisted cross breeding, that we are focusing to develop a kind of variety which are really climate resilient, those who can be grown very successfully under the drought condition, under acidic condition, soil conditions, submerged condition. So all these things are taking place, and I see that very bright future of this Indian Society of Genetics and Plant Breeding. And our secretary has also given a brief account of the achievement of this Indian Society of Genetics and Plant Breeding Ranchi chapter. I think this is fourth message. The secretary we are organizing since 2012, and I think this activity will continue under the blessing of Present Honorable Vice Chancellor, who also happens to be a plant breeder, and the research which are being undertaken and that has to be undertaken, that has to be given a kind of message that we have to increase our productivity per unit area and per unit time. And similarly, sir, just for natural resource management, as I said, the 20th century was devoted for crop improvement, but 21st century is only for natural resource management. This is the agenda or consultative group of international agriculture research, CGIR, and also ICR, because we are working with the National Agriculture Research System, and there are two partners of National Agriculture Research System. One is ICR, Indian Council of Agriculture Research, and another is State Agriculture University. So this is the agenda for 21st century for agriculture scientists, that we have to produce more and more in less and less. What is more and more food and more water, and what is less and less land and less water. This is the, I think, agenda for 21st century. With these few remarks, I really extend my thanks to inviting myself to be here today in this 2 days national seminar. Thank you very much.
Thank you very much for the beautiful presentation of the things going on in Jharkhand. As well as it is agriculture is concerned, we cannot talk about the state or the city. We have to talk about the whole nation. और बहुत ही खूबसूरती के साथ बेहद बेहतरीन तरीके से आपने कृषि पे रोशनी डाली है कि झारखंड में और जिस तरीके से कृषि हो रही है और आने वाले वक्त में कृषि के क्षेत्र में बेहतरीन आयाम क्या क्या स्थापित किए जा सकते हैं, उन पर बहुत बहुत धन्यवाद आपका. As we all know that we are here for discussing something better for the future, and we are waiting for the farmers to do better in their fields. We cannot wait for the natural resources, we cannot wait for the rain and other things which were the primitive way of agriculture. So now we are thinking of the modern way of agriculture where technology is very important. And for that, we need to get connected to the people who are working in this direction. We need to be nationally collectively working together for the betterment of the country. As far as any national seminar or workshop is concerned, people share their knowledge and experiences with each other. There are some learning people who have worked a lot and they know a lot, and it is very important to hear them and listen to them and follow their footsteps. So at present also, we have some person today with us. He is the guest of honor. He is the guest today. He is the president. I mean to say that he is the president of ISGNPP, president of Indian Society of Genetics and Plant Breeding, and he has also been working with the Ranchi chapter continuously and is today with us, and he is going to share his knowledge. So now we have the president, Indian Society of Genetics and Plant Breeding from New Delhi, Dr. G.P. Singh Sahab. May I request him to please be with us, share his knowledge and experience with us, with the Ranchi chapter and the other people who are connected in this national seminar at this occasion. So now I am inviting at this occasion the President of Indian Society of Genetics and Plant Breeding, Dr. G.P. Singh Sahab. सभी लोग कनेक्टेड हैं और हम प्रतीक्षा कर रहे हैं कि आपके सबसे तक हमारे साथ इस सोसाइटी के प्रेसिडेंट इंडियन सोसाइटी ऑफ जेनेटिक्स एंड प्लांट ब्रीडिंग के प्रेसिडेंट हमारे साथ मौजूद रहेंगे। मुझे से आज वो हमारे साथ कनेक्ट होने वाले हैं। कोविड-19 की वजह से सबकी मौजूदगी संभव नहीं है। हर एक व्यक्ति वहाँ मौजूद नहीं रह सकता ऑनलाइन कनेक्टिविटी से, और सभी लोग आज ऑनलाइन मौजूद हैं। रांची चैप्टर की सोसाइटी ऑफ जेनेटिक्स एंड प्लांट ब्रीडिंग के सदस्यों के साथ आज उनकी मौजूदगी में वो अपने रिपोर्टिंग के साथ हमारे साथ मौजूद रहेंगे। आज के अवसर पर
प्रेजेंट करना देखो तो इसमें अपलोड करना उनका साउंड अनम्यूट करना माइक हो गया फिर बोलिए हो गया सर नहीं ले लीजिए इसमें दे लीजिए माइक का फोटो होगा उसकी। हाँ सर, हाँ बस हो गया, छोड़ दीजिए। चला गया। हाँ वही बोल रहे सर, वो चला जा रहा है आपका सर। तो वही दिखाऊंगा मैं। वही बोल रहे सर, वो दूसरे लैपटॉप में कनेक्ट किया है सर। एक बार दूसरे लैपटॉप में कनेक्ट हो जाइए सर। जरूर उसकी बात करते हैं। हमारे साथ से कुछ नहीं। वो एक बात मुझको देखा
As we all know that we are connected online and due to COVID-19, everybody cannot be present here. Due to difficulty, there is some technical issue with the person we wanted to hear right now. He is the president of Indian Society of Genetics and Plant Breeding. We will be hearing him after this. Whenever we get connected to him, we will be hearing him. So everybody wants to hear him, and definitely as soon as we get connected to him, we will be listening to him. His views are very important, so we will be hearing it today. ये
तो ऐसा हो सकता है कि जो कनेक्टेड नहीं है उनको छोड़ के जो कनेक्टेड है उनका कर दिया जाए।
as per the program we are proceeding forward. जिनका कनेक्शन इनका नहीं हो पाया टेक्निकल रीजन से, उनकी भी बात हम जरूर सुनेंगे। जैसे ही टेक्निकल प्रॉब्लम सॉल्व हो जाता है, रिजॉल्व हो जाता है, we'll get back to him. So not wasting the time and taking in consideration the time. And everybody is sitting here to listen to the people who are eminent personalities who have enough knowledge, and we want to share with them. आज के इस अवसर पर जिनका वार्ता हम सुनना चाहते थे, वक्तव्य वो हम प्रेसिडेंट हैं, उनकी बात हम जरूर सुनेंगे। हम आगे बढ़ते हुए आज के इस अवसर पर, जैसा कि आप सब जानते हैं कि आज के इस अवसर पर हमारे साथ हम जिस विषय में बात कर रहे हैं वह कृषि में जेनेटिक्स एंड प्लांट ब्रीडिंग के महत्व के विषय में। जेनेटिक्स एंड प्लांट ब्रीडिंग का महत्व अपने आप बढ़ जाता है जब हम नैसर्गिक खेती से आगे बढ़कर तकनीकी खेती की तरफ जाकर और बेहतरीन उत्पादन के विषय में बातें करने लगते हैं। और ये सोने पर सुहागा तब हो जाता है जब आपके साथ एक ऐसा व्यक्ति आपके मार्गदर्शक प्रदर्शक और अभिभावक के रूप में आपके साथ मौजूद हो, जिन्होंने अपनी पूरी जिंदगी इसी फील्ड में काम किया है और इसकी बारीकियों से वाकिफ हैं। इसकी बारीकियों के साथ उन्होंने जिंदगी का सफर शुरू किया और इसी की बारीकियों के साथ आज वह एक ऐसे पद पर आसीन है जहां वो लोगों को दिशा निर्देश दे सकते हैं। उस व्यक्तित्व ने अपने जीवन के इस सफर में कृषि के क्षेत्र में बेहतरीन कार्य ही नहीं किए बल्कि कई जगहों पर मार्गदर्शन किया। भारत के विभिन्न हिस्सों में रहे और जेनेटिक्स एंड प्लांट ब्रीडिंग के विशेषज्ञ होते हुए उन्होंने इस क्षेत्र को बहुत ही बेहतरीन तरीके से समझा है। आज के इस अवसर पर इस नेशनल सेमिनार में हमारे लिए बड़े सौभाग्य की बात है कि वो हमारे साथ हमारे अभिभावक के रूप में हमारे साथ आज के इस अवसर पर मौजूद हैं। बिरसा कृषि विश्वविद्यालय कई सालों से निरंतर रांची चैप्टर के माध्यम से इंडियन सोसाइटी ऑफ जेनेटिक्स एंड प्लांट ब्रीडिंग के अपने कार्यों की चर्चा यहाँ पर करता है और अपने तरीके से बेहतरीन कार्य करता रहता है, जिसके बारे में हम सब हमेशा बात करते हैं। लेकिन आज एक सुखद अवसर है जब इस नेशनल सेमिनार में हमारे साथ माननीय कुलपति के रूप में मौजूद जेनेटिक्स एंड प्लांट ब्रीडिंग के भी एक एमिनेंट साइंटिस्ट हमारे बीच मौजूद हैं। हम सब प्रतीक्षा कर रहे हैं कि हम उनकी बातें सुने, तो मैं बिना विलंब मंच पर सादर आमंत्रित करती हूँ माननीय कुलपति बिरसा कृषि विश्वविद्यालय डॉक्टर O.N. सिंह साहब को।
Most respected Professor B.D. Singh, who has been Professor, Director and Director of the Banaras University. My colleague, respected Dean Agriculture, Dr. J.D. Haider, Associate Dean and Head Chairman, Department of Plant Breeding, Dr. D.N. Singh, Dr. Sohan Ram, Dr. वधूर डायरेक्टर रिसर्च DBG, और रजिस्ट्रार डॉक्टर साई, डॉक्टर PK सिंह, और डीन फॉरेस्ट्री डॉक्टर Bhatt, and all my colleagues from this university, respected Dr. GP Singh, my friends, and President of Indian Society of Genetics and Plant Breeding, who is also Director of Indian Institute of Wheat and Barley Research, my many friends from different part of the country, like Dr. S.R. Das, who has been known for his wide contribution, different kind of variety for different ecosystem in rice, and then many more like Dr. Kuldeep Singh, Dr. AK Singh, who is director of our very prestigious institute, rather one of the best institute in the country, that is Indian Agricultural Research Institute, USA, and then Dr. Banga. Perhaps he was here also. He was not doing this thing, but it is very good that he has been our colleague from Birsa Agricultural University in the long past time, and many more. Friends, we have to recall our glorious history. Like in every agriculture university in the country, if you just see, I am starting from my Pandagar, if you see anywhere, you see at least few breeds were known for their contribution, and their variety bred by them is still history in the country. If you take Sarju 52 from Eastern UP, that is Dr. Maurya, like that, Dr. S.K. Mohanty from Odisha, and Swarna, many more varieties. But somewhere we have to review our effort, the efforts of breeder. Why the present day varieties which are being bred not getting durability for a long time? Even within 5-6 years, they are not, just they are getting, I cannot say flat, but somehow durability is not like those varieties. Even if you see Varna, developed by Dr. D. Singh from Kanpur, still most of the seeds are from that variety, Varna, whatever variety you take from us. Like that, there are many more varieties. Still, Swarna developed in 1982, but still it has, wide coverage. Eastern UP, Bihar, many parts of the country, if you go, that will find that variety still in the pharmacy. Why I am doing it? We have to review of the effort. Why? So like that, the Birsa, Dr. Prasad will not mind, but somehow, the variety developed here in our university, why we are lagging behind? We do not have variety which has even 0.5 million hectare area developed in our university. I think Dr. D. Singh will not mind my comment, but somehow, as far as rice is concerned, somehow we are lagging behind. I was asking Dr. Prasad that when you will retire, he was telling that 2024. I do believe that certainly at least one variety will come which will be known in the future with his name. Like that, I was in the morning thinking that what should I speak instead of write, something written. I thought that I should speak something different than what we are very used to speak here and there. So I just wrote in my WhatsApp that a variety like Dr. Murli Dhan, you all know, because in DR back, he wrote in 2009 that there was very less genetic gain of rice for last 20-25 years. Although the rice production increases due to availability of better resources, including availability of yielding hybrids, this may be due to lack of the focus by the breeders for field evaluation and involvement of farmers in their breeding process. This also indicates that our national testing program, that is ECRIP, should be strengthened, particularly in the monitoring and evaluation. Study also shows that there is lack of seriousness. Please do not mind, I am just speaking from the core of my heart. Should be strengthened, particularly in the monitoring and evaluation. Study also shows that there is lack of seriousness in nomination for the testing by the breeders. The mega varieties like Swarna, developed in 1982, and few others are yet to be replaced, which were bred in more than 30 years before. It has also been observed that there is increased emphasis on introducing gene to a few mega varieties. Why we are not taking so many light races are there, so many, wild rice, and there are genes are there. Why we are not concentrating on those varieties? So that is why this is very good, but simultaneously, efforts should not be lessened to develop parallel mega varieties. Otherwise, our future generation will certainly blame us. We are taking advantage of our old breeders, but what we are going to put history, and that is also, please be serious. And many a time, being good friend Dr. Krishna Prasad, I have become little critical, so he will not mind. But before his retirement, at least one variety like Sir 52 or Dr. D. Chaudhary, he was from Motihari, Bihar, and he is known in every household in Odisha. The variety is Puja. Any household you go in Odisha, you will find his name Puja. They may not be knowing the name of Dr. Chaudhary, but they know his variety, that is Puja. Every family. So like that, I think Dr. Prasad will come out with one variety of that kind of variety. So friend, this I just wrote in the morning. Apart from that, it's a program, so I would like to speak. At the present stage, considering the importance of the topic and COVID-19 status of the country, it has been organized digitally, that is online webinar, that enable all interested participants without any risk taking advantage of the digital facility. The seminar is divided. I really congratulate Dr. Haider for meticulously planning. The various topic that is divided into 6 thematic area. Based on the importance of the different themes. That is one that is very important. That is what especially I was calling in the morning. Adaptation and yield stability. The success of a variety depends not only on higher yield. Why it is replaced? It is not that the variety developed by our present breeder, they are not high yield. They have yield, but it is not the yield only the farmers consider. They consider many more things, and that you can select variety unless you go at the farmer's field, you test on the demonstration. Please, whatever variety you are, when it goes at the advanced stage, almost near to release, when the varieties are in the yield testing, please at least in the stage of the 3rd year testing, please take it in the farmer's field also, try to take, so that the farmers can say that yes, if you are going to release this variety, it will be really worth to test. So please take it with the. Fortunately, our university has a very good extension system. KVC Vigyan Kendra, so many, almost every district we are having. So those should be tested at the farmers' field also before finally release the variety. So it is very important. High-yielding varieties should have wider adaptability. It means phenotype should be near to genotype, almost near to genotype. Then only can be stable and durable. We can say that reduced variation in the performance across location called adaptability, whereas stability refers to the reduced variation in performance across the years. Breeding program aims to develop a wider adaptable variety. It is essential to know the genetics of the trait, the mechanism leading to the development of the trait, and finally method of the measurement of the trait. Friends, for whom we are working, they are who are our clients. We have to understand this thing, and what is need of the clients, we have to understand this thing, and that is very important. Many a time we do hard work. There is no doubt. Everybody knows that Dr. Prasad. I am speaking name of Dr. Prasad because I was in his friendship for last more than about 15 years, I think. So that is why I am taking his name, and I have full confidence that he will come out before his superannuation. So. Sometimes we do not focus on the actual things where we have to identify what kind of trait we have to identify, what farmers require. So all those things we have to think, and that is only possible. Or maybe I request Director Research and Director Extension both that there should be a joint kind of interface meeting. I think every state in Odisha also there was an interface meeting with the help of the state government. Universities should conduct one interface meeting, and Director Research and Director Extension along with the different KVK and state government, please, my Secretary Agriculture should also be taken into consideration, along with all those team, like Director Agriculture and other directors also. So interface meeting, we have to, of course, I am deviating from the actual topic, but this is very important. Just today morning when I was thinking all those aspects, I thought, why not we are also conducting. What about Wadul Sahab, whether we have conducted the interface meeting earlier? So please let us start interface meeting.
Yeah, we used to have some. When I was director research, I used to.
So let us again take it, because it is very essential, and this will help to stabilize the variety. So all the breeders, please take a note of it. Before taking the variety finally released, please test at the farmer's field also through interface meeting, because it's a different team. So there are many more things to. I will be more listener than telling more things. Then plant genetic resources and pre-breeding. Pre-breeding is nothing but germplasm enhancement. How, because there are many good genes in various wild type of the you can say not variety exactly, but there is wild type of the many crops. In the rice also are many more crops. There are wild kind of genes, and they can be trapped and they can be taken into the adapted variety, which are very useful. So we have to think on those areas also. That is basically germplasm. Pre-breeding is nothing but germplasm enhancement. Like that, climate change. Nowadays, everybody is looking climate change. So we have to think for climate smart variety, which can suit in different variety environment. Like when there is more of the rain, when there is drought, when there is, so we have to think for multiple stress. And of course, few varieties in the past, few variety have been developed, but the requirement Dr. D.L. Singh in the morning, in his talk, he was telling that revolution. Normally, we are used to speak about the wheat and rice, but there are many more crops like sugarcane. He spoke, maize. He spoke. Of course, one thing, the potato also. There is huge improvement in potato. So I am talking about the crop only. Otherwise, if you see the milk production, that is also many times, maybe 5-6 times more compared to 50 years back. Fish also 5-6 times more. Then even the pulse also. Huge production is pulse also. So like that. But still, oil seed, Sohan Ram, oil seed still we have to work hard. Countries importing huge oils, especially edible oil. So and last almost 2.5 years I was in plant variety protection appellate tribunal, and the variety developed by the breeder, because unless we give importance to breeders, unless we give them the right kind of intellectual property right, nobody will invest in R&D of the varietal development. So that is why I am very much interested that breeders should come out. I will give all kind of support if they go for, and I request Director Research. I think he is also doing formal release of the variety.
Yeah.
So please take it, and at least in the first or second month of the next new year, this meeting should be conducted. Please take time from the Secretary Agriculture, so that at least few variety we develop. Release. Already there the material has been developed in the pulse also, the linseed also, I think, rice also, Dr. Prasad. So, the thing is not only to work hard, we have to come at the result also. The problem is that many times we work hard, but we do not come at the end point. So the plant breeders right, farmers right, and you see in the whole world, the importance have been given only to breeders, but India is the only country where as far as plant intellectual property is concerned, farmers were given equal kind of right. Because if you think, many variety which have been identified and developed by the farmers that are still in the field. So government to incorporate farmers' claim also, farmers' right also. That is why our, PPVFR, that is Protection of Plant Variety and Farmers' Right Act. Only India where the farmers were given so much importance. In not other country, only plant breeders were given importance. So that is, and the credit, much of the credit goes to our Dr. Professor Swamina. So then, right of the nowadays you cannot take variety and develop by breeder, like maybe Professor Singh or maybe Neeraj or anybody. You have to take permission. Only for one time you can take variety, but repeated use of the variety, you have to take permission from the concerned breeder if you want. Not that you can take and then you are going to take and then you can go for commercialization of that material. You have to take the written permission of the person concerned who has developed the original variety. So all those things are there. Then, so my only message is that why I am speaking all those kind of things. The breeders are not devoting enough time in the field. So I was very harsh in Karnataka also as head of the department. I was putting more pressure. So please devote at least considerable time in the field also. Otherwise, your variety development will not be durable. It will be quickly. I mean, the farmers will not take because of you have developed variety. Farmers will take only if they are feeling its importance for their own. So please devote enough time. Field testing is very, very important. It should be rigorous. Why variety developed at Marmateru and Bapatla compared to any part of the country, they are more stable, adaptable, and successful. Because of course, geographical location is also one of the factors. But the people, I mean, who are there, they are giving enough time in the field also. And I am not criticizing friends. I am telling this. I mean, kind of our young generation, they are not giving enough time in the field also. Of course, the supplementation of biotechnology is equally important. But it is the time, maybe after 50 years, biotechnology may complement, but the time which we are running, that is only maybe supplementary, not complementary. Biotechnology has to supplement, but we have to devote enough time in the field. And only then, you see, I was seeing in my earlier days, the breeders were at least 3-4 hours in the field, they were devoting time. But somehow, I mean, this is not only for Birsa, but this is for the whole of the breeder fraternity. Please devote enough time in the field. Do not only by going at 10 in the field or just 10-15 minutes and returning from the field, it will not give you enough. And see, sometime I will call Dr. D. Chaudhary here. You will just listen from him. Today also Dr. Sardas. He is a very successful and accomplished breeder from OUT, Odisha University of Agriculture and Technology. He will give our lecture. So please listen him very carefully, and I request all my breeders from this university, please listen him very carefully, and then you will learn. And he is very accomplished breeder. Thank you so much for all your effort. I especially Dr. Azhar, his whole team who took pain, and perhaps I am very lucky that it was planned in March, but somehow it is being conducted after I was here. Thank you, sir. Thank you so much for your meticulous plan and all those things. Thank you.
Thank you very much, sir. As we all know that we were trying to get connected to the president of ISGNPP, Indian Society of Genetics and Plant Breeding, and we really wanted to hear him. हमें बहुत खुशी होती है उनको सुनकर, लेकिन तकनीकी खराबी की वजह से हम उन्हें नहीं सुन पाए, और मैं तहे दिल से आज यहाँ पर इस अवसर से क्षमा मांगती हूँ कि हम किसी टेक्निकल फॉल्ट की वजह से आपसे कनेक्ट नहीं कर पाए। जरूर फिर कोई ऐसा अवसर आएगा जब हम आपकी बातें सुनेंगे, आपके विचारों से अवगत होंगे और आपके अनुभव का लाभ ले पाएंगे। हम पुनः एक बार विश्वविद्यालय की तरफ से आपके आभारी हैं कि आपने इतना समय दिया, और हम आपसे क्षमा प्रार्थी हैं कि हम आपके पास नहीं पहुंच पाए। हम कोशिश करेंगे कि हम आपके पास जरूर पहुंचे और आपके अनुभव से हम कुछ लाभ ले पाए। थैंक यू सो मच फॉर बीइंग विद अस एंड थैंक यू सो मच। हम सब जानते हैं कि माननीय कुलपति महोदय हमारे आज के इस कार्यक्रम के मुख्य अतिथि हैं, लेकिन आज के इस कार्यक्रम सम्मेलन की जो अध्यक्षता कर रहे हैं। मैं यूँ कह सकती हूँ कि जेनेटिक्स एंड प्लांट ब्रीडिंग के सिनोनिम के रूप में जिन्हें पूरे भारतवर्ष में जाना जाता है। आप ये कह सकते हैं कि वो पर्यायवाची हैं। जेनेटिक्स एंड प्लांट ब्रीडिंग का हर एक विद्यार्थी जिस तरह माँ का हाथ पकड़कर चलना सीखता है और सबसे पहला कदम अपने घर के आंगन में डालता है, ठीक उसी प्रकार जेनेटिक्स और प्लांट ब्रीडिंग की तरफ जब भी आपके कदम बढ़ते हैं तो वो पहला नाम जो आपके कानों में गूंजता है वो है डॉक्टर वी सिंह का। वो न सिर्फ जेनेटिक्स एंड प्लांट ब्रीडिंग के सभी लोगों के लिए मार्गदर्शक है, बल्कि उनकी किताबें संपूर्ण भारत में सभी लोग उसे अपने संविधान की तरफ पढ़ते हैं। तो जेनेटिक्स एंड प्लांट ब्रीडिंग के सिनोनिम के रूप में हम जिन्हें जानते हैं, वो व्यक्ति जिन्होंने इस क्षेत्र में बेहतरीन कार्य कर अपने आप को, अपने व्यक्तित्व को लोगों के बीच में ऐसा कर लिया है कि लोग उनके बिना जेनेटिक्स और प्लांट ब्रीडिंग की कल्पना ही नहीं कर सकते। सभी के लिए बहुत ही आदरणीय आज हमारे साथ इस कार्यक्रम में अध्यक्ष के रूप में मौजूद हैं। इस कार्यक्रम की अध्यक्षता कर रहे हैं। अध्यक्षीय संबोधन के लिए मैं आमंत्रित करने जा रही हूँ डॉक्टर वी सिंह फ्रॉम BHU। Presidential address के लिए may I request for the presidential address Dr. V.K. Singh from Banaras Hindu University.
क्या हुआ?
हम देख लिए थे।
Good afternoon.
आप ही का लॉगिन थी, हम आपके लैपटॉप में घुस गए हैं। जी इसमें आवाज दे रही है।
Good morning. Can you hear me?
Yes sir.
Should I begin?
Yeah.
Okay. Am I visible there?
Yeah sir, visible.
Okay. Good morning, everyone. I'm very happy to join you and to express my appreciation for all concerned with organization of this seminar, particularly the Vice Chancellor, Birsa Agricultural University, and the teachers of the Department of Plant Breeding and Genetics to be able to organize the deferred seminar during December time. And before I begin the presentation that I would like to share with you, let me apologize for being late in joining you, as a plant biologist, I have been trying to catch up with the technology in breeding and genetics, but the electronics technology has beaten me very rapidly, and I am outdated on that. That is why some of the problems were there. So I would like to begin my formal presentation now. Present your entire screen. Okay, share. Dr. O.N. Singh, Vice Chancellor, Birsa Agricultural University. Mr. Ashish Kumar Padhi, CGM NABARD. Dr. G.P. Singh, President, Indian Society of Genetics and Plant Breeding. Professor M.S. Yadav, Professor D.N. Singh, Professor Z. Haider, and Dr. Sohan Ram, distinguished participants, dignitaries in the audience, young plant breeders, ladies and gentlemen. It's my privilege to share some of my thoughts on the gene editing and the draft guidelines for gene editing developed by DBT. Gene editing is a technology that started in 1996. That was the first paper published on it. It started moving by 2011 and caught up speed in 2013 in the real sense, and is one of the most intense areas of research in plant biology today. What is done in gene editing and how it is done, I'll share with you. But let me first point out that it is very enabling technology. It can achieve everything that mutation breeding can do and that transgenic technology can achieve, only in more efficient and highly targeted manner. The three things it can do is inactivate genes, it can replace the alleles of selected genes, and it can integrate transgenes at selected genomic sites. The tools that are used for gene editing are called site-directed nucleases, or SDNs. The first nuclease was zinc finger nuclease. This is based on transcription factor zinc finger proteins and is very difficult to design for new targets. In 2011, the second nuclease, site-directed nuclease came about that is called TALEN. This is based on tail-like effectors that are produced by pathogenic bacteria like Xanthomonas. And they are really easier to design, much easier to design than zinc finger nucleases, but they are still difficult. The darling of plant biologists or those working in the gene editing arena is this RNA protein nuclease, CRISPR-Cas9. This is the most liked nuclease for one simple reason, that the nuclease action is provided by Cas9, which is a protein, whereas the selection of genomic site is done by an RNA that pairs with the DNA. An RNA being a copy of DNA, a transcript of DNA, is very simple to design for new targets. Actually, you all need to design is only these 22 nucleotides to rearrange them that pair with the genomic target. The mechanism of action of site-directed nucleases is like this. The nuclease is taken to the selected site by its DNA pairing component, where it induces a double-strand break. And this double-strand break is repaired by the cellular DNA repair enzymes. Generally, this repair will be done by non-homologous end joining, in which case the two ends of the broken DNA, they are joined together. But during this process, there is either random deletion or random insertion of some nucleotides. And because of this reason, about 2/3 of the products of repair will lead to frame shift mutation and inactivation of gene. The double strand break also enhances homology directed repair several thousandfold. Therefore, if the researcher desires to integrate or copy at the break site some DNA sequence, he has to prepare what is called donor DNA and provide that along with the nuclease so that the sequence is copied at the site of the break, like here. The changes that are brought about in the genomic location where break occurs are classified into three groups: SDN1, SDN2, and SDN3. In the SDN1, as I said, the repair is done without any DNA being provided. And this repair leads to insertion or deletion of few to several bases. Therefore, the products that are generated by SDN1 technology, they are identical to the products of mutagenesis. The problem, there are three slight difficulties with this, with any gene editing technology, and this is here also, that there can be sometimes the low frequency of changes that you did not want to occur at the targeted site. There may be also changes in very low frequency at sites that you did not target. And sometimes foreign DNA may become integrated into the genome, particularly if you are using DNA copy of the SDN for transformation. The DPT requires that the intended genetic change is confirmed, the presence of off-target site is ruled out, and phenotypic performance of the mutant is shown to be equivalent to that of the parental line. And also, a more important, foreign DNA should be absent for the genome, from the genome. The regulation is required to be done at the level of institutional biosafety committee. The global scene is that in USA, Argentina, Brazil, Australia, and in some European Union member states, SDN1 is considered to be a non-regulated article. The DBT proposal for regulating SDN2 is like this. In this technology, sequence within a gene is changed by the sequence that the experimenter wants to insert in the genome. By this activity, you can change a mutant allele to wild type allele. You can change an existing allele to another allele that is present in the primary gene pool. Both these types of changes will produce products that are identical to back cross products, actually very clean back cross products. As a result, these two kinds of replacements should be exempted from regulation. They are exempted from regulation in Argentina and USA. The DBT there is another kind of change that can occur. That is, you can replace the sequence within a gene to create a new allele that is not known to be present in the primary gene pool. Consequence would be that the function of this allele will not be known, and possibly this regulation should be, this product should be regulated, but the level of regulation should be much lower than conventional GM crops. The global scene for regulation of this kind of product is not very clear. What DBT proposes is that the targeted gene editing is confirmed. There is no biologically significant off-target editing. There is absence of foreign DNA, and assessment of phenotypic equivalence, trait efficacy through appropriate contained and are confined field trials. It's not very clear what the DBT wants here, but possibly they want trials materials to be handled just as GM crops are handled. The regulation will be based on performance tests, and they will be approved by Genetic Engineering Approvals Committee. The GED3 or SDN3 technology will integrate transgene from any source into what is considered as a safe genomic site, a genomic site where transgene integration will neither activate nor inactivate any gene that you do not want to activate or inactivate. And it will also not lead to position effect on the transgene expression and stability. The project will show effects of the transgene that has been moved into, and in my opinion, this is what should be evaluated. In normal GM crops, there is problem because of random integration in the genome of the transgene, and that phenotypic effects that are unpredictable. The suggestion is that the regulation should be at a lower level than for conventional transgenics. The DVT proposes, however, to regulate SDN much in the same way as it did GM crops, and the approval would be by genetic engineering. Approvals come in. The global scene on SDN3 is not very clear because not many products have been developed. NAS held in February a roundtable discussion, and after comprehensive discussion, it was concluded that SDN1 and SDN2 should be exempted from risk assessment and regulation. And their field evaluation should be done under ICAR, All India Coordinated Project System, much as the conventional bred crop varieties are evaluated. And the release and notification also should follow the same procedures and protocols that are followed for normal crops. The proposals of NAS are being considered by DBT. The last I understand is that they are debating about the SDN1 regulation. To conclude this, the global impression is that if you consider SDN1 and SDN2 as regulated article and wish to enforce their regulation, it will be extremely difficult to separate them out from varieties that are produced by conventional bycross or by mutagenesis. Therefore, this will be left to the honesty of the developer of the variety to declare that it is produced by gene editing. Otherwise, if there is a strict regulation enforced and it becomes very difficult to get through varieties produced by gene editing, then there would be encouragement, or it may lead to encouragement of dishonest practice, because the developer will be far safer to say that these varieties are conventionally grown. In Argentina, I should note that the policy of regulation is proactive in that the authorities advise any breeder, any company, or any institution interested in following gene editing project or any other kind of project, they advise them at various steps what kinds of regulatory procedures or hurdles they are going to face, and also proactively advise how they can be avoided, those hurdles. Maybe someday we should think of developing a system like that is friendly to breeders and developers of gene editing varieties. In the end, and this is not a plant breeding situation, I should like to share 3 quick slides on human welfare, particularly centric to human species. There is a simple genetic device called Gene Drive. This has the CRISPR-Cas9 CRISPR-Cas9 system. Any gene that the experimenter wants to spread in the population, And this construct is flanked by two sequences that are homologous to the sequences present at the site in the genome where you want gene drive to be inserted. Um, the what happens in case of gene drive is that if there is a gene drive heterozygote produced, the CRISPR-Cas9 will be produced, and this will cut the homologous wild-type chromosome at this site and repair by homology-directed repair of the double-strand break will lead to copying of the gene drive into the other chromosome. So the heterozygote becomes homozygote, whereas normal Mendelian transmission will maintain proportion of the mutant phi or gene drive phi to the same proportion generation after generation, unless there is selection. Where in case of gene drive, this goes on spreading in the population, and in about 13, 14 generations, the gene drive will replace, virtually saturate the population and replace the wild type allele. What effect this can have? Just two, I'll cite that you can control a pest population like mosquito. And as a matter of fact, British firm is testing this gene drive in Brazil for control of Aedes aegypti mosquito. Gene drives will be spread in any crossbreeding population. So there is another type of thing. An extremist group could develop a gene drive that affects a beneficial crossbreeding species and spread the gene drive in the population of that species, and the beneficial species may become extinct. So you can eradicate uh, a nasty species. You can rescue a beneficial species if it is endangered. You can also eliminate, uh, if you are extremist, you can eliminate a beneficial species. Uh, in laboratory, these gene drives have been demonstrated to work very well, but their application is not yet allowed, except for very specific situations. Finally, to all those who are interested in human health, gene editing has been used to develop some gene therapies that are useful in curing very serious diseases, cancer, leukemia, and so on. And the the one that I would like to focus on for just for the advertisement purposes, the last one, Zolgensma. This is for an incurable disease, unmanageable disease that means definite death to the child in 2.5 years age, spinal muscular dystrophy. The the gene therapy is the costliest drug on earth. One dose for full treatment costs $2.1 million. Uh, Many gene therapies will be developed. One gene therapy that concerns many individuals who require T cell replacement is the development of what is shown here in the first one, CAR T cell therapy. Normally, T cell will have to be isolated from individual patient used for development of the modified T cell before it is introduced. But gene editing has been used to produce universal T cell that can be taken out and used with any patient. It can be used anywhere. And this CAR carries um, carries a gene that identifies a cancer-specific receptor, binds to it, and kills the cancer cell. So this T cell therapy is targeted to specific cancerous cells and leads to their elimination. Very precise, a magic bullet-like situation. Uh, I hope I have been able to explain to give you an idea of gene therapy, its potential in changing genotypes of crops, particularly, as I said, humans, because we are interested in human health, and also it would be able to develop many more gene therapies for diseases that afflict us. I thank you all and wish the conference a great success. I would like to again specifically thank the Honorable Dr. Owen Singh, the Vice Chancellor of Birsa Agricultural University, and the teachers and employees of the Faculty of Agriculture, and particularly Department of Plant Breeding and Genetics. Also, I would like to appreciate the technical support that was extended by Dr. C.S. Mato and his team, and thank you very much.
Thank you very much. Thank you very much. We had Dr. V.D. Singh from BHU and we were listening to him. It was really a privilege to listen him today. जैसा कि हम सब जानते हैं कि इसके पहले हम कनेक्ट करने की कोशिश कर रहे थे हमारे आज के इस कार्यक्रम में हमारे प्रेसिडेंट ISGNPB या इंडियन सोसाइटी ऑफ जेनेटिक्स एंड प्लांट ब्रीडिंग के प्रेसिडेंट हैं, तो हम जरूर उन्हें सुनना चाहेंगे। तो एक बार और कोशिश हम कर सकते हैं उन्हें कनेक्ट करने की। अगर आज वो कनेक्ट हो जाते हैं तो हम उनका भी वक्तव्य और बोधन आज यहाँ सुन पाएंगे। He is the president of the Indian Society of Plant Breeding and Genetics, so we would like to listen him and we are trying to connect him once again.
कनेक्ट हो रहा डॉक्टर जी को कनेक्ट करो, कनेक्टेड है, बात कर सकते हैं वो।
So we are not able to connect him again. Thank you very much to him for being with us today and bearing this technical fault from our side. Thank you. Thank you for being with us. And we are sorry for the technical fault. Now, as we all know, that we are in the national seminar and this national seminar is going to continue. We are going to further discuss the important issues and things which will be giving benefit to the farmers and the society after the discussion. So for that, we will be moving further for the sessions. But right now, in the inaugural session, we have to thank the people who have been with us at this occasion. So for that vote of thanks, may I invite Dr. Somanraj.
Thank you, madam. Most respected Dr. Owen Singh, Honorable Vice Chancellor, Birsa Ranchi, and Chief Guest of inaugural session of the webinar on crop breeding for wider adaptation. President of the Ranchi chapter of Indian Society of Genetics and Plant Breeding, Dr. B.D. Singh, Dr. G.P. Singh, President of Indian Society of Genetics and Plant Breeding, New Delhi, Dr. M.S. Yadav, Dean, Faculty of Agriculture, Dr. D.N. Singh, Vice President of the Ranchi chapter, Dr. J.D. Haider, Secretary, Indian Society of Genetics, Ranchi chapter, Dean PGS, Dr. M.K. Gupta, Director Research, Dr. Madhu, Registrar, Dr. M. Kudada, DSW, Dr. D.K. Sahi, Dean Forestry, Dr. Siddiqui, Associate Dean Gadwa, Dr. Pal, Dr. Chaudhary for MBBGR, Chief Scientist Dr. Manigopal, faculty members, participants, students, press and media. Proposing a vote of thanks is solemn task. The task becomes more solemn and pious when it is on an occasion of webinar on a very relevant topic, crop breeding for wider adaptation. I feel pleasure in proposing a vote of thanks on behalf of myself as well as on behalf of Ranchi chapter of Indian Society of Generation Plant Breeding and Birsa Agricultural University. First of all, I would like to thank our Honorable Vice Chancellor and Chief Guest of the National Seminar, Dr. Ranchi, under whose dynamic leadership, guidance, and direction this seminar is being organized. A special thanks to Dr. B.D. Singh, a renowned scientist of the country and president of Ranchi chapter, who has been guiding the chapter for meeting the objective of the chapter. In absentia, my sincere thanks to guest of honor Sri Ashish Kumar Padik, Chief General Manager, NABARD, Jharkhand. I thank Dr. G.P. Singh, a dynamic president of Indian Society of Genetics and Breeding, who has always helped this chapter in this academic pursuit. Thank is also to Dr. M.S. Yadav, a vibrant in agriculture, who is always inspiring and ready for extending perspectives that are at his. I would like to thank Dr. D.N. Singh, Vice President of the Ranchi chapter, for all positive support to the cause of the society. A special mention for Dr. J. Heather, who is a person responsible for all such activities, not only of the chapter, but also for departmental and university. I am thankful to deans, directors, registrars, head of the departments, scientists, students for their participation. I am thankful to persons from press and media for covering the program, and their critical and analytical coverage is highly acknowledged and appreciated. Last but not least, I wish to record the contribution of the member of the different committees, especially Sri Ajay Kumar, Mrs. Punata Kujur, and Ms. Fatima Nath, who meticulously managed the compilation of abstract, and Sri Dhananda Raman for online webinar management. Thanks all. Once again, thanks. Jai Hind, Jai Jharkhand. Thank you.
Thanks a lot for the vote of thanks. Now may I request all of you to please stand for national anthem.
पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव
So now we were connected online for the national seminar on crop breeding for wider adaptation, offline and online. The seminar's inaugural session was going on, and after this, for a break, we will be returning for the technical sessions. So now we are going offline at present. In the technical sessions, we will all be getting online connected again. अभी हम ऑनलाइन जुड़े हुए थे, लेकिन आज के इस कार्यक्रम में हमारे साथ मुख्य अतिथि के रूप में माननीय कुलपति बिरसा कृषि विश्वविद्यालय मौजूद रहे। उनकी गरिमामयी उपस्थिति यहाँ आज के इस कार्यक्रम में थी। मैं अनुरोध करूंगी हमारे बीच मौजूद आज के इस कार्यक्रम के नेशनल सेमिनार में हमारे साथ ISGPV के सेक्रेटरी डॉक्टर जडे हैदर सर से कि आज के इस अवसर पर हमारे इस कार्यक्रम के मुख्य अतिथि माननीय कुलपति बिरसा कृषि विश्वविद्यालय को शॉल उड़ाकर उन्हें सम्मानित करें। सर आगे
बंद रहते हाँ कपाइलू छे
ये नेशनल सेमिनार